thank you for inviting me. I've not worked with organic phosphorus before, and I'm really learning a lot now. I worked with the inorganic fraction, and more specifically on the rocks. All phosphorus originally come from rocks, so that's a good starting point. So this is a rock of 4 billion years old in Canada. One of the few that survived because they weather and they get recycled and they go into the organic fraction, all the elements. It's a very long title. I've been working on fungal weathering for quite a long time, and it's the same as with all research areas. You get a lot of different ideas and they stay there and people tend to believe them, but you really have to test them, all those ideas, and put them in context. So that's the mineral, that is fluoroapatite. It's full of phosphorus and calcium, and there's the fungus and the tree. There's a picture I got from Håkan Wallander, and I really like that picture. And it is the interaction. So why working with apatite? Apatite, it's phosphorus is an essential element, a nutrient for the trees. And what you see in the lab, if you have a microcosm and put grains of this apatite in there, and you have mycorrhizal hyphae going there, and they really go for the apatite. They recognize it, they like it, and maybe they do something with it. Yeah, I don't know if all people are familiar with mycorrhizal fungi. These live in symbiosis with plants, and I'm focused on ectomycorrhizal fungi. They live in symbiosis with many coniferous trees and also a lot of broadleaf trees. And I have a picture later on here. First ID, the picture will come later. First ID, what is going on? And on the left, you have this organic P, so that's all the organic P in my talk today. The idea that you have a hotspot of a resource, and then you have a root with your mycorrhizal fungi around it. So the fungi, they get a lot of energy from the tree, they produce enzymes, and they can get the phosphorus out. And the idea was the same happens with rocks. So those rock fragments, they contain nutrients, but they are really locked up. And there you have the tree roots with the mycorrhizal hyphae, and they go to specific minerals, and they produce acids and siderophores, other compounds, and try to retrieve nutrients from the minerals. So that's the idea. But what do you need? You need the transfer of this plant energy to specific minerals. A lot of the minerals is not interesting for plants. You have a lot of quartz in the soil, and that doesn't contain any nutrients. So there needs to be a specific located transport. The next one, if the fungus is there, it should be able to take up nutrients out of the mineral and bring back to the plant. So there must be a plant access. And then the last one, that's a lot debated, but by getting access to it, so you take up elements from your mineral, so you increase your dissolution rate of your mineral. But how much does it account to your total weathering rates? And it can be on different scales, on the fungal scale, on the ecosystem scale, and even on the global scale. And there are a lot of papers, mostly review papers, dealing with that. And on the global scale, you have a modeling paper by Taylor et al. But that's modeling, so it's based on these ideas. And what is needed is more evidence, more data on those fluxes. Okay, first back to this picture. So that was my inspiration, and when I started in Sheffield, we made microcosm systems and we worked with different minerals, and it was only with apatite that the fungus really went for it, they really liked it. So we started more sophisticated systems. And here you see a picture of the mycorrhizal root tips. So you have a tree root here, and then the mycorrhiza, they force the tree to produce these very nice root tips, and they envelop the root tips, and in that way they have a very intimate contact between the tree and the fungus. And what you see here, they're all fungal hyphae going into, these are mineral pads, but also into the whole system. So this is a very interesting infrastructure, and you will find it in the soil as well. So that increase the uptake capacity of your tree a lot. And for phosphorus, that's really important because it's not really mobile. So what we did then was we put in boxes or little caps with different content. So here we put in quartz sand with a little apatite, and in that one as well. And in these two we just had quartz sand. And you can't see it from here what's going on, except you see a little bit the root structure is going in that way, but we used 14C labeling, and there you see that all that plant energy that's fixed that goes into the apatite wells. So what you see here on the single grain scale, you see here on the patch scale. So there is a direction of plant energy towards those patches. And of course the question is it driven by simply the dissolved P in those systems? You have apatite that will start dissolving, so there's available phosphorus there, and the fungus sense that and bring back phosphorus to the tree and get more energy and going to explore that? Or do they sense the grains as a potential source? And I think that's an essential question, because in more natural systems, you have your grains, individual grains, patch over your whole matrix. There's not much of an increased available phosphorus level. So another approach to look at this transfer of plant energy is, so that was in the microcosm system, and I like to go back to the field because that's where it is going on. And then I went back to my my PhD work. I looked at thin sections. I really like this technique. So you have a soil sample, you fix it in a resin, and then you make a microscope slide of it. And then what I did was I stained different minerals, so I could recognize different minerals. I stained the hyphae. This is not a stained sample, but here you see the dark septated hyphae, but there are also many hyphae that are not visible just with normal light. So you need to stain for that. And then I quantified how much of the surface of each mineral was colonized by fungal hyphae. For 3 different minerals, for quartz, for potassium feldspar, and a plagioclase. Plagioclase is a sodium calcium feldspar. And what you can see, this one, the plagioclase, is more colonized than the other one. And I didn't put any statistics here, but it's highly significant, as you can expect. So also on the field scale, there is a specific translocation, at least of fungal biomass towards different minerals. But then you have to realize those quartz grains were dominating your soil matrices. It was around 80% quartz and then 20% of the other minerals. So in total, most of the fungal biomass was still on quartz grains that doesn't contain any nutrients. Another approach is what we did when I was in Lund with, in the lab of Håkan Wallander. We had these mesh bags filled with different grains, different minerals. We placed them in the forest, and it was an interesting site in Czech Republic. Close together we had different bedrocks. So we had the granite, we had the serpentinite. Serpentinite is really high in magnesium and really low in phosphorus. And then amphibolite was high in everything. And then we looked at different minerals. So we had mass bags of apatite, with biotite, with hornblende. Hornblende is also a magnesium mineral, and with just quartz. For most of the minerals, we didn't see any effects on the colonization, and we quantified colonization with acosterol. Acosterol is a measure of fungal biomass. But what we can see, if the ecosystem is very low in phosphorus, you see an increase in apatite colonization. So here again, there is a tendency of increased colonization of apatite. And this is a mass bag, so I don't know if we put 1% apatite in there or something like that. So it's probably higher than what's available in the soil. So it is a hotspot. But anyway, the fungi recognize the phosphorus source. So I think there is the first idea is we have enough evidence to see there is some specificity in the soil, in the direction of the plant energy. Of course, it's the question, is that enough to expect really increased weathering rates and on plant nutrition level enough effect on your trees? I doubt that, but that's the discussion for later. So then we go to the next one, the plant axis. So back to these systems, very simple microcosms, and we could do a mass balance because it's a very clean system. And what you can do is we had systems, we didn't add phosphorus, of mineral phosphorus, so in the nutrition in the agar on the bottom, and ones where we added phosphorus as well to sufficient levels. If you add a lot of phosphorus, the tree take up much more phosphorus. They were not phosphorus limited compared to the other systems. And then we had this quartz, or we added apatite for both treatments. And what you see here, without any phosphorus source, there were very poor trees. They survived, but nothing more than that. So this is just the background phosphorus from the agar and from the quartz sand. And then when we added the apatite, you saw a strong increase. That's this part. But most interestingly, there's a lot of increase is in the root system, and that does include all the fungal hyphae. What you see here is probably that a lot of this phosphorus got stuck into the fungal biomass. But at the end, you see also an increase in your shoot phosphorus. So there is part of the phosphorus from the apatite ends into the plant shoot. So there is a transfer of phosphorus. Okay. I'd like to go back to the field again, and other people did it. When I did my PhD, this paper came out, a famous paper in Nature. I think it's, what was it, 2002, I think, or 2001. With this title, mycorrhizal weathering of apatite as an important calcium source and base for forest ecosystems. So this is based in northeastern USA, and those forests can be quite low in calcium. And apatite has, of course, calcium, and it weathers much faster than other calcium minerals, so it can be a potential source. And the work was based on measuring calcium-strontium ratios and strontium isotope ratios in the different fractions, in different soil fractions, in tree leaves, through fall, through the canopy. And if you look in detail, then you see, so these are separated, those three species. So these are the leaf data from all the other leaf data. And this is the apatite. So it's getting very close to the apatite. So the conclusion is those trees, they get a lot of the calcium from the apatite. And because apatite is distributed randomly into the soil, there must be a direct access bypassing just the soil solution to it. And that is what the authors concluded. Ectomycorrhizal trees take up calcium directly from apatite. And when I read this as a PhD student, I thought, it's all very interesting, but what about the other ectomycorrhizal trees? So the spruce and the fir, the ectomycorrhizal, but birds and beech as well. And they appear here, very close to the non-ectomycorrhizal trees and the ferns, and very close to the exchangeable calcium pool. So to me, it's not an ectomycorrhizal effect, it is an evergreen effect, and it's really big risk to work with element ratios because of fractionation. That if you have a tree species, an evergreen tree species with a very long-lasting leaves, where you can get accumulation of calcium oxalates in the leaves, and you get leaching out of calcium from there, you can get fractionation, and that's known for a very long time. I came across a paper, I think it was from 1919 somewhere, so quite a long time ago, and they already warned for the use of calcium-strontium ratios, and then this was about blood and using oxalates in blood samples, but the same is true here, and probably in those plant leaves, calcium oxalate plays an important role in the recycling of your calcium. So about the direct access for plants, I think there is evidence from the microcosms, but we have to be very careful. But about the field, I don't think we have any evidence that plants can take up specifically from specific minerals directly elements. And of course, it's really hard to prove. So then we go to the last point. How to quantify weathering rates? That's a big challenge because it's such a slow process. That's a project, that is an experiment I did during my PhD. It was my first experiment. I came in there as a student in a project called Rock-Eating Mycorrhizas, and that was all based on the finding that on certain minerals, again this is a thin section, you can find structures that look like tunnels, and in some tunnels you find hyphae. And the idea is that those hyphae really penetrate, mine into certain minerals. And this is a feldspar. So my first question was, okay, very interesting, but how much does this contribute to total weathering? My supervisor, Nico Verbremen, he thought that must be 45, 50%. I don't know what he mentioned in the papers, but really high. And then after some work, some half-year work, I think, I was a little bit disappointed. It was less than 1% contribution. And then I continued working with more fancy methods to see can we find interactions of those fungi with minerals on the fungal level. And in Sheffield, I worked together with a lot of people. We worked with atomic force microscopy, and what we saw, if we had a very flat biotite flake, a mineral, and the fungi growing over it, you saw these little trances, and you could see the same morphology as hyphae grow. So you see some interactions, and we also made cross sections with FIP. So on focused ion beam, you can drill into your mineral, and there's a fungus growing on top of the mineral. So there they are growing, and then you get the cross section, and then you can use TM, EDX, so you can use other fancy methods. And what you see close at the surface, so you're talking about nanometers, You see a depletion of potassium. We also saw an increased oxidation of your iron. So you see things going on. But again, the big question is for these observations, how much does it account to the ecosystem scale and to the forest nutrition scale? So I wanted to work more on the ecosystem scale. So I moved to Lund to work with Håkon Wallander. He knew of a site in Norway, and that's a very interesting site. So this is an old picture. We went back and we had a color camera. So, but first, the site, it's a very gentle slope, and on top there's a sandstone that is leaching lead. So you have a lead gradient that causes a vegetation gradient, and here you can see when we visit the site. And this gradient is probably already there since the end of the glaciation, because that was the geological setting that was left after the ice sheet retreated. And that's a very interesting gradient to look at a weathering over a period of 10,000 years. So what we did, we recognized different vegetation types. So you had the bare soil, you had the grass, you had some poor trees that tried to survive. Then with the bigger forest, you had a border where vaccinium didn't want to grow and started to grow. So they called the area where we have just the complete forest to control at the border that we called vaccinium. Then we had the poor tree, then the grass, and then the bare soil. And then we took soil samples to look at the apatite. So you have a vegetation gradient, you probably also have a microbial gradient and a mycorrhizal gradient. So that's the ideal system to test our hypothesis. And our hypothesis was that those mycorrhizal fungi, mycorrhizal vegetation has an effect on your apatite weathering. So first we quantify apatite content, and what you can see, there's a much more apatite left on the bare soil than in the control. And also there is a depth effect on the surface, you have less than deeper. We also measure pH. And why did we measure pH? Because pH is a major factor in your dissolution rate of your apatite. Now we got a lot of data. We could look like here, so you have your different, so this is the deeper soil, and then the lighter the color, the more on the surface. And how you want to look at it is if you look at the kinetics, here you have the pH, and then you have the dissolution rate, and in the acidic part you get a straight line. And that is very useful if you want to analyze your data. And that's based on those kinetics data. And this N is important. I will show you later. So what we could do was to plot for each soil depth a straight line. And then if we looked at the deepest soil samples, 30 to 40 centimeter, that N of this function, this kinetics function, is almost exactly the same as what you find in batch experiments. So there was an indication that at that soil depth, it's a proton-driven weathering. And on the upper soil layers, something else is going on. Now, of course, you think, there must be then a fungal weathering going on top of your proton weathering. So we measured acosterol in it. And then to our surprise, with increasing acosterol in the top layers, so the light ones, you see a negative relationship. More acosterol means less dissolution. So that's opposite than what we expected. There's a lot of noise there. And then on the deeper soil layers, you see the opposite, but that is really noisy data. Put together, so pH, acosterol, they could explain 90% of our weathering rate. And I thought that is, with such a simple model, it's quite nice. So I thought, what is going on? What is causing this fungal inhibition of weathering. And then I had something in my mind, or studies from a long time ago, that humic acids can block dissolution rates. They attach to mineral surface, to the active sites, and they block the dissolution reaction. And then at this point, I started working with organic matter, and I knew that mycorrhizal fungi oxidize organic matter and producing a lot of dissolved organic matter. So wrapping together, so I measured the absorption at 280 nanometers, so that's a measure of your more complex phenolic dissolved organic matter. And I could make some correlations with acostrol, with 13C, and what I call the deviation from the pH model. So minus, then you have inhibition of your weathering rate, and on the right you have an increase of your weathering rate. And what you can see here, with higher organic matter content, you have inhibition of your weathering reaction. And at the end, I think what is going on, you have your fungal activity. They could increase your apatite dissolution by specifically going on those minerals and produce acids. But you also, on the same hand, you have the production of dissolved organic matter, and that hinders the dissolution rate. And this just to show that if you can focus and study phenomena on a very isolated scale, but at some point you have to go back to the whole system. And I think they apply for all mechanisms that go on in the soil. And just the last slide, just to wrap up. So we have to take care of, take into account there is this transfer of energy towards specific locations. So soil are heterogeneous. Maybe there's some plant access, but I don't believe it has a major effect on your plant nutrition. And the weathering rate, there are local effects, but I don't think it's on the bigger scale. And it's not just based on this data, but also on the kinetic theory. And by that, I like to thank all the workers at the different universities I've worked, and I'd like to thank you.